In Chapter 22, we're talking about nucleic acids. We'll start out by talking about what makes a nucleic acid, and being able to identify the differences between DNA and RNA. We'll then move on to talking about primary and secondary structure in nucleic acids, and then we'll talk about how DNA is replicated and how DNA is eventually translated into protein. We'll also talk about things that can go wrong with DNA, like mutations and viruses. Our fourth type of biomolecule is a nucleic acid. Nucleic acids include DNA and RNA, and even though nucleic acids make up a very small percentage of our overall biomass, they play an outsized role in the function of our cells because they contain all of the genetic information that is eventually translated into proteins, the largest bioorganic component of our cells. DNA is a type of nucleic acid. A nucleic acid would be any polymer whose units are nucleotides. Of course, nucleotides then need to be defined as well. Those are any molecule that's made up of ribose, which is a carbohydrate, phosphate, and some sort of nitrogen heterocycle, a ring that contains nitrogen. We can see those pieces at play in our different nucleotides. On the top, highlighted in purple, you see the different types of bases that are present. Remember, we had talked about the structures pyrimidine and purine in Chapter 17 as being particularly important because those are the structures that were present in DNA. So you can see the top row in purple are all pyrimidine bases. They all have the 2 nitrogens in the aromatic ring. And the bottom 2 are adenine and guanine, which are purine bases. They have the 4 nitrogens in the 2-ring heterocycle. Those bases will all be connected through something very similar to an acetal bond to either a ribose or a deoxyribose. A ribose for RNA, a deoxyribose for DNA. And each of those will be connected to a phosphate or multiple phosphates through a phosphoester bond at the final alcohol position, at the 5 position. In both DNA and RNA, we have 4 bases that we use to encode all of the information that we use in the nucleic acid. However, the bases that we use for DNA and RNA are different. In both DNA and RNA, we use both purine bases, adenine and guanine. In both DNA and RNA, we also use cytosine, one of our pyrimidine bases. However, the other pyrimidine base is different depending on whether we're in DNA or in RNA. In DNA, we use thymine. In RNA, we use uracil to accomplish the same maintenance of information. Notice the difference between thymine and uracil is only the CH3. So other than that CH3, those bases are identical to each other. DNAs are really just a short way of saying deoxyribonucleic acid. Deoxy meaning that the ribose itself is deoxygenated, and ribonucleic acids would then be just the straight ribose for the carbohydrate. We can see this difference in the deoxygenation if we look at RNA and DNA side by side. If we look at the RNA structure, we'll see that the ribose that is used for RNA has an OH on the 2nd position. In DNA, that OH is replaced by a single H. That would be the deoxyribose that is used in DNA. Again, we see that thymine and uracil are different, while all the other bases are the same between DNA and RNA. We also will talk about the possible nucleotides a little bit different for RNA versus DNA. We will abbreviate them with a 3-letter code. The first letter code of the code refers to which base is used. The second letter, the M in this case, refers to how many phosphates are present, and the P in this refers to that there is phosphates. So for AMP, it stands for adenosine monophosphate. GMP would be guanosine monophosphate, etc. If we had ADP, it would be adenosine diphosphate, and ATP would be adenosine triphosphate. Notice that's a little bit different for our possible nucleotides for DNA. They always come with a D in front of them indicating deoxy. So then we have deoxyadenosine monophosphate or deoxyguanosine monophosphate, and so on. While certain nucleotides, like adenosine triphosphate, are relatively ubiquitous throughout the cells, many of our nucleic acids actually come as polymers. Each of those polymers are going to be linked by the sugar, by either the ribose or the deoxyribose. So the basic structure of any nucleic acid is going to be phosphate linked to a sugar, which is linked to the next phosphate, which is linked to the next sugar, which is linked to the next phosphate, and so on. So for DNA, it would be phosphate linked to deoxyribose, since that's the only sugar, to a phosphate, to deoxyribose, and so on. Whereas in RNA, it would be phosphate linked to ribose, linked to phosphate, linked to ribose, and so on, since ribose is the sugar that's used. Notice that in either of these cases, the base that is used is not part of the backbone. It's always a side chain, much like the side chains of amino acids are not part of the backbone. The primary structure of the DNA and RNA is going to be linked together through those phosphoester bonds. We also talk about these nucleic acids with a distinct directionality. We always list them from the 5' end to the 3' end. That's because that's the direction that they are synthesized in our body. The 5' end would be the end that has the 5' open. Notice that the 3' is where it's linked to the next carbohydrate in that 5' end. And the 3' end of the entire nucleic acid would be where we have an open alcohol on the 3' position. When we link all these together, the phosphates are always going to be connected as diesters, and they always carry a -1 charge, which makes nucleic acids really quite anionic. They're really quite water soluble. The backbone is always going to be the same in every one of these cases. Just like we talk about the sequence of amino acids as the primary structure of a protein, we also talk about the sequence of our nucleotides as the primary structure of our nucleic acids. Our DNA and RNA also have secondary structure on top of the primary structure. RNA secondary structure is widely varied and relatively difficult to understand. It's outside the bounds of this class in most cases. DNA secondary structure, on the other hand, is relatively straightforward and important for how our bodies maintain consistent information across many generations in our genes. DNA will automatically arrange into a double helix, which is shown on the left. And in the double helix, 2 strands of DNA will be held together by hydrogen bonds between complementary strands. Notice in the structure on the very left, there's a strand that it goes from top to bottom, starting at 5' and ending in 3'. The complementary strand goes in the opposite direction, starting at 5' on the bottom and going to 3' at the top. For this reason, we talk about DNA complementary strands as reverse complements. They're going in opposite direction and they are complementing each other. When we say the strands complement each other, we mean that every time we have a certain base pair, it always has a pair that goes with it. Those pairs are determined based on 2 different factors. In order for the DNA double helix to maintain the same distance between the helix at any given point, we always have to have the same size base pairs. So we'll always have exactly 1 purine and 1 pyrimidine, so that they take up the same amount of space. We could have 2 different forms of T, in this case, pairing with a purine. So here we have T, which is a pyrimidine, pairing with A, which is a purine. And here we have T, which is a pyrimidine, pairing with G, which is a purine. Notice that when T pairs with A, there are 2 hydrogen bonds that connect these 2 bases. That's a much stronger interaction, and so T will always match up with A. We say that T and A are complementary. Similarly, C, which is a pyrimidine, can either match up with G or with A. When C matches up with G, we end up with 3 hydrogen bonds between those, whereas when we match C up with A, there's only 1 hydrogen bond. The C and G matchup is much, much stronger, and because of that, we end up with C always being the complement for G. Knowing what we do about DNA complementary strands, If one strand is given, we can always know the other strand is in the reverse direction and complementary. Take a look at the first strand shown and try to identify what the reverse complementary strand would look like. The second strand would go from the 3' to the 5' direction. Reading from the 3' direction, there would be an A underneath the T, a T underneath the A, and A, T, C, G, A, T. So for every T is paired with an A, and every C would be paired with a G, but the strands would be in opposite direction. Now pause and try to identify what the second strand would look like. Again, we see every C would be paired with a G and every T with an A, but again, the 3' end would match with the 5' end and the 5' end with the 3' end. The strand would be in the opposite direction. Because our base pairs have exact matches, these strands can always be separated and we can synthesize the reverse complementary strand and create new DNA. The matching always is going to take place from the 5' end to the 3' end by an enzyme called DNA polymerase, which matches the direction that we write our DNA in. Notice that if we're trying to create DNA from 5' to 3', that works really well when we start at the 3' end and work the opposite way. It doesn't work as well in the complementary strand, and so the complementary strand is created in piecemeal segments called Okazaki fragments that are later attached together. It's pretty difficult to separate all the hydrogen bonds that hold DNA together, so it happens just a little bit at first, and as complementary strands are synthesized, it becomes easier and easier to pull apart the DNA as it replicates. We call the places where the DNA starts to pull apart origins of replication, and eventually, as the origins of replication spread, we end up creating 2 strands of DNA from the single template. This whole process is called DNA replication, and it creates 2 exact copies of DNA from the one original DNA molecule. These DNA strands make up our chromosomes, which are complexes of the DNA and protein that together store our genetic information. It's one thing to copy our DNA to store our genetic information, but it's another thing entirely to actually do anything with that. While DNA is able to maintain steady sources of genetic information, RNA is the route that we actually use to copy that information into a useful form as proteins. We call the copy it from DNA to RNA transcription. The RNA will eventually carry that information to our ribosomes to be translated into proteins. We already have talked about 2 different major differences between DNA and RNA. Try to remember what those are by looking through your notes. Here are some other ways that DNA and RNA are a little bit different. RNA is almost always single-stranded, and because RNA is single-stranded, it cannot error check in the way DNA is. It's not a good long-term storage for genetic information. RNA is much smaller than DNA, and RNA tends to have many different functions, and we'll see many of those functions through the transcription and translation processes. There are 5 major functions of RNA that we'll see throughout transcription and translation. The first of these is heterogeneous nuclear RNA, which is formed directly from DNA transcription. hRNA would be a direct copy of a gene on the DNA, the gene being a segment of DNA that codes for a specific protein or group of proteins. We oftentimes talk about the genome, which is the entire set of genetic information. It's the collection of all available genes. So the heterogeneous nuclear RNA, it comes directly from a gene and is copied. But the heterogeneous nuclear RNA is not converted directly into a protein. It actually first gets sliced into smaller pieces so that only the useful information is brought. The smaller pieces are messenger RNA, and the small nuclear RNA are what help to cut the hnRNA to the mRNA. We'll also see during the process of translation, the ribosomal RNA is required to actually form the ribosomes, the enzymes that allow us to create proteins. And the transfer RNAs are going to actually do the translation, the interpretation of the RNA signal into proteins. And we'll talk in more detail about how each of those work. The first step of this process, transcription, is actually really similar to DNA replication, except that now we only need to make one copy for the RNA. We don't need to make two copies, and the DNA will rewind rather than separating into two strands. This means the hRNA is produced all by itself, and the DNA is maintained as permanent storage of that genetic information. After the hnRNA is produced, the hnRNA is converted into mRNA. The hnRNA consists of two types of pieces, both exons and introns. Exons are the types of pieces that will actually eventually give information for a protein, and the introns are unnecessary pieces for that signal. Those pieces, the introns, need to be removed, and the small nuclear RNA are the ribozymes, like enzymes but with ribonucleic acids, that are able to remove those sections. When the exons are joined together, they become a messenger RNA. Now the hRNA are copied from genes, but not everything that is in a gene is turned into a protein, and not every protein from the gene is the same. We can take a set of 4 exons, like is shown here, exon A, B, C, and D, and we can actually splice them together in many different ways. We don't always have to include all of the exons. That means that many times we can create more proteins from a single gene than just a single protein, which means that the transcriptome, the complete set of all mRNAs that can be produced from a genome, is actually much larger than the genome itself because of all the possible alternative splicing patterns. Now that we have messenger RNA, we need some way of taking the RNA nucleotide sequence and converting it into an amino acid sequence. To do that, almost all organisms use the same universal genetic code, a matching of 3-letter sequences to specific amino acids. In general, don't memorize these codes. However, I do want you to know what the start codon is, or the initiation codon. In RNA, the initiation codon is AUG, which matches an ATG sequence in DNA. Those codes are interpreted through transfer RNAs, or tRNAs. tRNAs are bound in the ribosomes so that the anticodon section, a 3-letter sequence on the transfer RNA, is able to match on to the codon section of the messenger RNA. Each of those transfer RNAs has a specific site to attach an amino acid that is specific to that transfer RNA. The 3-letter sequence of a transfer RNA anticodon is able to match up with a messenger RNA codon in such a way that transfer RNAs will line up next to each other and bring the amino acids that are attached close to each other in order for them to react in sequence as well. But how do the amino acids get on the transfer RNA in the first place? We accomplish these with specific tRNA synthase complexes, or enzymes, that are able to take a tRNA that matches with a specific binding site for that enzyme and bring it close to a specific type of amino acid that gets charged or loaded onto the transfer RNA. After the RNA is transferred on to the amino acid, or the amino acid transferred onto the RNA, it can bring the amino acid to the ribosome to be synthesized. The ribosomes are actually enzymes that consist of both protein and RNA that together perform the translation of the proteins. So ribosomal RNA is part of the ribosome enzyme. Ribosomes consist of both a large subunit and a small subunit that both work together. Because the ribosomal RNA plays such an important role in this, we call it a ribozyme, an enzyme that is an RNA. This is actually our second example of a ribozyme, the first being our small nuclear RNA, which helps to cut our heterogeneous nuclear RNA into the messenger RNA. Proteins at the ribosomes are translated in 5 steps: activation, initiation, elongation, termination, and then post-translational processing. We'll walk through each of these steps in the next slides. Activation is where the tRNA synthase attaches an amino acid to a tRNA. This actually happens outside the ribosomes, but is required to constantly be happening while enzymes or protein synthesis occurs so that we have a steady supply of charged tRNAs. After activation, the mRNA will be bound to the small subunit of the ribosome at an AUG sequence, remember that's the start codon, in the messenger RNA. A matching tRNA is going to bind and bring along an amino acid, in the case of the start codon, a methionine, and a large subunit along with it. So now we have a single tRNA bound into the ribosomal complex at what we call the P site. The next step is elongation. That's the process by which amino acids are added to the chain one by one, each time forming a new amide or peptide bond. After the first tRNA is bound, a second tRNA binds according to the next three base pairs, making sure that its anticodon will match the codon in the messenger RNA site. As that bond happens, a new bond is formed between the first and second amino acids, and the first tRNA is then released. The ribosome moves down the mRNA and binds onto the next tRNA. The process continues over and over, each time elongating the chain of amino acids by one. At some point, we need our protein to stop being formed. Termination is the process by which elongation stops and the protein is released. It occurs when specific stop codons, UAA, UAG, or UGA, don't memorize those, appear. There are no tRNAs for these stop codons, so they signal protein release. Post-translational modification can now occur after the protein is formed, or even actually while the protein is being formed in some cases. The types of post-translational modifications that we see include things like cutting off the first amino acids. Oftentimes the methionine is not required, or other amino acids are produced that help the protein to fold, but then are later cut out. Disulfides also form at this time, helping to stabilize the tertiary structure of the proteins. And anytime we have quaternary structure, we need multiple peptide units to come together, which means it has to happen after we've produced multiple peptide units. Many ribosomes can participate in the translation of one mRNA at once, so the ribosomes can actually bind at multiple locations along the mRNA, which means that we can use a single mRNA to make many, many copies of a protein. Now, we've talked about this before, but we have 5 major functions of RNA, and now we've seen how each of these are used in the processes of transcription and translation. Heterogeneous nuclear RNA is the first copy that's made. Small nuclear RNA cuts that copy into messenger RNA, which is actually the piece that will be translated into protein. The piece is translated in the ribosomes, which consist largely of ribosomal RNA, and they're interpreted by transfer RNAs that bring along the correct amino acids to the translation sites. We've talked a lot about how replication, transcription, and translation work when things are going right. Some things do not go right, and mutations are an example of that with DNA. Mutations are errors that are made during DNA replication that result in the changing of genetic information. Usually having 2 copies that are complementary results in having an ability for our cells to check whether there are errors, and when they relatively rarely happen, they're usually fixed. Sometimes the DNA is actually changed. A common example is when a mutagen is going to change the structure of a gene. Substances can be mutagens, but we can also have UV light and other ionizing radiation be mutagens. When a mutagen changes the structure of a gene, that then changes the proteins that are produced from this. And usually this is a bad thing. Positive mutations are fairly rare. So we rarely get things like Teenage Mutant Ninja Turtles or superpowers. It's a lot more common that we get something like cancer. Another way that DNA or RNA can end up going wrong is in viruses. Viruses are small particles of protein and DNA or RNA that need a host to replicate. They don't have any of their own central metabolism. This creates huge problems for our immune systems because the viruses are going to inhabit our cells themselves and use our own central metabolism. We treated a lot of pathogens with inhibitors that can target specific aspects of the pathogen central metabolism, but when the central metabolism is our own, we can't kill off the virus without killing off our own cells. When viruses insert their genetic material into host cells, the host cell itself is forced to express the genes contained in the viruses. The only way to really proactively deal with viruses is with a vaccine. Vaccines are dead or weakened viruses that allow our immune systems to learn to recognize them and start to fight them off at very little risk. Vaccines have to be given before exposure to the virus. They do almost no good if they're given after the virus is contracted, which is why it's essential that vaccines are done proactively as recommended by doctors. A good analogy for how viruses work is found in the section 3rd matrix, if you've seen those by any chance. Agent Smith functions as a virus in these movies. Agent Smith cannot create copies of himself without having a host within the matrix that he can insert his code into. And I think the best place to end any chapter is with the discussion of Agent Smith in the matrix.